My project is on the phototransduction of the rod cells in the retina. We all use phototransduction every single day, even though most of us have no clue we are doing it. This is how every single individual sees light and dark. This picture shows a simple example of the human eye. Here we have a pupil, the retina, and then the cornea. The light goes into the eye and hits the back of the retina. The retina is broken down into two different parts. These are the rods and the cones. The cones allow us to see all of the beautiful colors that surround us, and the rods help us to see light and dark. A good way to remember the difference between cones and a rod is that traffic cones are a bright color, like orange, and rods are usually dark. The main thing that is located in the rod is rhodopsin. This molecule has two separate parts called opsin and the retinal. When light is exposed to the retinal, it changes from a curled up line to a straight one. This results in rhodopsin also changing its shape, and then it becomes a part of transducin. The transducin has three separate parts to it: the alpha, the beta, and the gamma. Since the rhodopsin changed its shape, it makes the alpha part of the transducin break off and become part of PDE. PDE's purpose is to break down cGMP. When both cGMP and PDE combine, it then transfers into GMP. This transfer results in the sodium-gated channels to close because there is no more cGMP floating around to keep them open. Since the gates are closing, positive sodium ions can't get through, and if they can, only a few. The cell then hyperpolarizes, which makes the cell turn negative and then shrink. This concludes my presentation of the phototransduction of the rod cells in the retina.